Good day, class, and I hope you guys all had a good weekend. Today's lecture is going to be a little quick review of the main things I really want you to know about protein and enzymes. Enzymes are like the coolest proteins. They're the worker proteins. They do all the jobs. Make sure you're taking notes through this. There's going to be some vocabulary in this presentation. I would recommend going back to the PowerPoint version, not the video version of this, and just copying down those vocab words. Going to be really important you get those understood. Here we go. A little review of proteins. What are they made out of? Hopefully remember that they're made from amino acids. How many of them are there? There's 20 in the human body, 9 of which are essential. So, that's pretty good. Remember, they have different charges, polarities, hydrophilic, hydrophobic, and it helps with that folding of proteins. Okay, so here we look at the folding of proteins, the different stages of protein folding, and you get the sequence of amino acids is the first thing that's going to determine shape. And then depending on how they are, if these two have disulfide bonds, they'd connect here. If they're hydrophobic, it'll bend these in different positions, and you can get different shaped proteins. So kind of talked about those. Primary, remember, is single-stranded. Then we get our beta and alpha helixes. Those then form together to form a three-dimensional protein, and then multiple units of these, like 2 insulin proteins, form this, or 4 for hemoglobin, give you that quaternary structure. And that's your complete protein. Really, in truth, we'll learn later that some of those get shipped out into the Golgi, where like lipids are put on them, or certain carbohydrates are added to them as well, depending on their sequences. But that is the basic of proteins. Haven't got into this one yet, but this is our next big topic. This is when we'll get into genetic engineering as well. But explain how a cell knows how to make a protein. And it comes down to the DNA. This is the dogma of biology, and its DNA gets transcribed to mRNA in the nucleus. This mRNA then leaves the nucleus and actually joins a ribosome and amino acids in the cell to produce a protein. So this is the code for that sequence of amino acids. Pretty amazing stuff. We are going to spend a lot of time on here. I'm going to go ahead and skip over this part. I don't know why I put that there, but bring some ways a cell could explain that it's called expression of DNA. We'll get into that much later. So functions of proteins, we talked about them. You could be structure. Enzymes is what we're getting into today. They're my favorite. They're used for signaling from cell to cell. Receptors are part of signaling too. Channels to allow things to go in and out of the membrane. They help us move. They help us transport things. This is the vocab I was talking about earlier. I would go back and just copy this down from the regular slideshow if I were you, but we'll go through them quick. A catalyst, this is going to speed up chemical reactions. Okay, so anything that speeds up a chemical reaction is a catalyst. I think you guys talked about that in chemistry probably quite a bit. The activation energy, that's going to be the energy required to cause a chemical change. So even if the chemical change is going to a gas, you have to put energy into it. If I want to break down sugar, I need to put energy in it. If I want to make sugar, I have to put energy into it. So activation energy is that chemical energy required. Substrate is going to be what the enzyme reacts with. So an example for this is lactose is the sugar that's in breast milk. Lactose combines with lactase, which is an enzyme. So lactase is the enzyme, lactose is the substrate. It combines to it. So substrate's what the enzyme's going to change, either put together or take apart. Active site is where the enzyme attaches to the substrate, and we'll get into that. And the product is what is made after the enzymatic reaction. So it's what the substrate will become. So in my example of lactose being a substrate, when it makes with lactase, it's broken down into glucose and galactose, which are 2 different monosaccharide sugars. Enzyme substrate complex is what we're going to call it if the enzyme and substrate are actually bound together. And allosteric site is something we'll talk about too. It's part of an enzyme that is not the active site. So I'll have you write those down. You'll want to know these as we go through what's going on. So just some definition of enzyme. Enzymes are proteins. They're the workers. They're awesome. They're like the tools. I like to think of them. If I was building a house, I'd need screwdrivers. I would need the drywall machine to hold drywall up. So any of the tools I need to build things or tear things down in a cell, those are enzymes. The definition biological form are proteins that catalyze a chemical reaction by lowering that activation energy for the reaction. So lactose, for an example that we just talked about, that breaks down into two sugars, it will happen naturally on its own. I had a 30-year-old box of lactose that I used to use for an experiment, and after 30 years, it had actually all turned into glucose. But it took 30 years. That wouldn't help your body out very much. So enzymes make it happen much, much faster. So almost instantaneous. And we'll do some labs to look at that. Big thing, they're like special specific tools, okay? So they're very specific for what they do. This is just a graph kind of showing how enzymes work. So here's the energy needed for the substrate to turn into the product. With no enzyme, we'd need a lot of energy would be used to get there. With the enzyme, we lower that by over half with this one. So just kind of get that idea going. Oh, okay, sorry for the pause there. I had people talking outside. I needed some water. I mean, trying to lecture at a computer is horrible. Big thing about enzymes, I was kind of getting into this, they're very substrate specific, okay? So one enzyme works on one substrate. You don't have an enzyme that works on two substrates. They don't do different jobs for each other. They all do the same job. That's really important because I don't want to have my enzyme that's breaking down sugar all of a sudden start breaking down my cell wall, or if I'm a plant, or my cell membrane if it's me. That would be really bad. So they have to be very specific, and we also have to be able to control them. Okay, and some of the specificity comes down to the shape. So this lock and key method that we'll talk about. So the shape of an enzyme is specific to the substrate. It's all about shape. Proteins are all about shape. So enzymes are a protein. It's all about shape. So they only work on one. And what's really cool is once they get done doing their job, like once this enzyme comes in and breaks something apart, it goes and starts working on the next one. So they get recycled, they get reused. It's not a one and done kind of deal. Enzymes can keep going until something denatures them and they go bad. Remember, we talked about denatures with proteins. At home, you guys can watch this lock and key animation. I think you're going to kind of get it from what we talk about. So we talk about something called the lock and key hypothesis. This is a very simple way to explain the specificity of an enzyme and how they fit like a physical lock and key. And this is kind of the idea of the lock and key hypothesis, is we have our substrate and it's specific to the enzyme shape and they're bound together. They bump into each other. These lock together, and that's how the enzyme works. This is a very simplistic method. I'm going to prefer to use the induced fit model. This is more accurate. It's a little more difficult to understand, but not really, so I don't even know why I talk about lock and key. Induced fit allows for, a little different things, like maybe something with a similar shape can actually fit in here. It doesn't have to be perfect, but similar might fit in here, and then it could stop enzyme reactions or something like that. So in this case, the enzyme change their shape slightly. So when they combine, there's actually a slight change of shape. So the charges are right where this thing kind of grips down on it. And that change of shape then will cause a chemical reaction in here that maybe puts these two things together. And we just had a anabolic reaction and we created something new. Maybe that change of shape breaks it apart. So they become bound to each other. And basically the idea of induced fit is you get a change in the enzyme shape to make things work. Okay. We're going to come back to this idea because like I was talking about, for example, cyanide poisoning actually fits into the active site. Cyanide fits into the active site that allows cellular respiration in us. And so when cyanide is here, it doesn't actually allow the electron to come into this enzyme and make ATP for you. And so you die because you have no energy. And it's because another chemical fit that similar shape. Drugs work like that a lot too. And then in other cases to regulate proteins, we'll talk about this too, is certain substances can bind to somewhere like here, and that can cause the change of shape here so it doesn't come in. I hope you guys can see my mouse on there. I actually don't know if you can. Last things I want to talk about quickly, and then we're going to do some labs on this in class, so we'll talk about this more in person as well. So this is kind of just a quick overview. The effects of temperature, pH, which remember is like acidic or basic, and substrate concentration on enzyme activity. And I think the best way to look at that is to look at a graph. So I'd like you to kind of explain this graph. So this could be any enzyme. I'm just using a basic example. But we have our enzyme activity, how fast it's working, and temperature. And so if you look at human, this would be a human enzyme. What would you say the optimal temperature for enzyme activity is for humans? Okay, and you'd come up, and that's pretty easy. 37 degrees Celsius, which is body temperature. That kind of makes sense. But I'd also like you to think about what happens to our enzyme activity as temperature increases. So from here down at 0 degrees to 10, it starts to get more and more activity. And the reason this is you have lots of enzymes. The picture of me, my fingers, I like to use my fingers. For an enzyme to work, let's say my cupped hand is the enzyme, this is the substrate, those two have to come together, okay? So they have to be going in the cell, they're moving around, and they have to come together before a reaction occurs. Well, when it's cold, things move really slow, and so there's less chance that they bump into each other in the right way so that they can have their reaction. But as temperatures go up, molecules start to move faster. If they're moving faster, they're more likely to bump into each other, and because of that, we're going to have more reactions occur. So as temperature increases, we get more enzyme activity until we reach this certain point where the enzyme actually becomes denatured because it's too hot, and then we get nothing. So it very quickly is going good, 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 and then dies. And that would happen to us if we got too hot, because we would die. This is a thermophile, so this would be a bacteria that lives in like the thermal vents of a volcano or something, or hot springs. And you can just see that their enzymes too increase with energy, but their denaturing point is a much hotter temperature, so they're able to survive in much hotter temperatures. Pretty cool life. I guess we have not talked about denaturation. So a quick review of denaturation. Basically, it means a protein's conformation or its shape changes, okay? So when a protein gets too hot or too acidic or too basic, it changes its shape, and once its shape's no good, it doesn't work anymore. So conformation is the change in shape of a protein, and denatured just means the protein cannot work anymore. I already talked about things that would denature it. Temperature, heat, definitely can break them up if it gets too hot. Those bonds that hold them, as that is vibrating, it just breaks away. You see that in water when it boils, like the little intermolecular bonds of water break away, it boils. Cold can sometimes denature, but oftentimes if cold denatures, it actually can recover after it, which is kind of nice. There's just heat denaturing the proteins in an egg. We're used to seeing that. pH can change it. So if you remember right, your amino acids have certain charges, so they have to be in the right pH to work correctly. So proteins have to be in the right pH. If that pH changes, bonds change, it denatures it, it breaks it. There's normally a range, like you're pretty good from say like, 4 to 8 or something like that. But if you drop down to a 3 or up to a 9, you can have some serious damage. An alkaline environment like bleach is really good at killing things because it's really good at denaturing proteins. While very acidic conditions, sometimes the protein can actually recover from that as well. So here's just some egg things too. So here's temperature denaturing. This was just done by increasing pressure. And here was just changing the pH. You can see it definitely changed that. So kind of cool. I would like you to define denaturation, and it's the structural change in a protein that results in its loss of biological protein properties. I'm going to end it there for denaturation, but just kind of an idea that that's why you see this huge dip off in proteins. So we didn't write it down yet. I have you to write down what we talked about with the effect of temperature on enzyme reaction. And remember, from here, the molecules are bouncing around faster. They're more likely to run into each other. So our enzyme activity increases, and then we hit the denaturation point and it drops off exponentially. went through that already. Substrate concentration. So this is the lab we're actually going to do in class. But if I have lots of substrate, there's more chance for it to bump in to the enzyme, right? Or if I have more enzyme, there's more chance for the substrate to bump into it. But we're going to talk about just how much substrate there is. If there's a lot, you can see as substrate concentration increases, our reaction rate really increases until you hit a point where it might flatten out here, and that's because every enzyme is being used. So at a point, your limiting factor might be how many enzymes you have, so you can't work any faster on the substrate because you run out of enzymes. But in any case, as substrate increases, there's more chance those enzymes have a substrate in it, and we get more reaction time. So the reaction rate increases because there are more substrates present, increasing the chance that the substrate will meet the active site. And because the enzyme is catalyzing as many reactions as possible, that's why it kind of plateaus or levels off at a high concentration. So that is substrate concentration. I want you to have an idea about this because when we talk about regulating cell processes, concentration of substrate is a really good way to regulate it. So if I have lots of substrate, it kind of slows itself down in this plateau, or if I have very little, it might speed back up. And this will be the end of our notes on enzymes. I'll have you guys watch a video as well, but try to explain this graph. These are two different enzymes. We have protease. This breaks down proteins, and this protease is located in your small intestine. And then we have lipase, which breaks down fats. So lipid, ACE, protein that breaks down fats. Lipase is a protein that breaks down lipids. Anything that ends in ACE is a protein. Is an enzyme. Sorry, enzymes are proteins, so that's not wrong. But what's going on with this is they have different optimal pHs. So you can see things work better at an optimal pH. The pH of your small intestine is about 8, and you can see the protease works really well at a pH of 8. Lipase actually works better at a 9, okay, according to this graph. So just pH, enzymes have specific pHs they work better at. Different enzymes have different pHs. Gastric lipase is a type of lipase in your stomach. Well, your stomach acid's a 3. So you'd actually see down here that works really well in a really acidic environment, where this one works better in a basic environment. That was a lot for notes. Like I said, I would go through that by hand, and we will do some of those in person as well. You guys have a great day. Don't forget to answer your questions, and I will see you in class unless I already saw you in class.